All right, so today we're going to talk about cells and the substructures within those cells called organelles. We're also going to highlight why cells are small, as well as talk a little bit about the cell theory, which is what we're going to start with, okay? So first thing you need to understand, cell theory. All organisms are made up of cells, okay? So the smallest unit of life is a cell. The smallest thing that can be considered living is a cell, okay? Some organisms are single cellular, some organisms are multicellular, a bunch of cells put together, but the smallest thing that can possibly be considered living is a cell. Okay, that's one thing. Also, cells come from pre-existing cells. So that means that cells will divide, and through a process called either mitosis or binary fission or budding, there's a bunch of different ones we'll get into later in the year. But that's all cells have to come from pre-existing cells. So that should beg the question, right? Where did the first cell come from? Things we'll talk about later in the year when we get to things like chemical evolution, okay? We talked about it's the simplest matter of collection of matter that can be considered living. And also, cells contain hereditary information. So that is, they have genes, they have your DNA within the nucleus of the cell. We'll get into that a little bit more when we start talking about that organelle specifically. So another important thing, just like we talked about the biomolecules, right? Structure, function, structure, function. The structure of the cell, what it looks like, the proteins it makes, is related to the jobs that it does. You have all of these different cells in your body. Each of them has the same genetic material. They have the same recipe book of genes, of DNA, yet they look so different and have many different functions. How does that happen? When we get into genetics, we'll talk about how different genes get turned on or off. It's called differential gene expression. So my muscle cells are not going to make the same proteins that my nerve cells are going to make. Some of them will be the same, but not all of them. And that's very important for what's called cellular differentiation, or having different types of cells, okay? So structure function relationships, again, not only in biomolecules, move it up to cells. We can even move it up to tissues, organ system, organisms, even the structure of ecosystems as well. So we see these themes throughout all the different levels of life. So first cells to exist, and these still exist today, are prokaryotes. Prokaryotes are things like bacteria. They are very simple. They're basically, and we'll talk about this in a second, you'll list it, they're DNA surrounded by a cell membrane. There's a bunch of ribosomes in there for making proteins. There's a cell wall on the outside. They have cytoplasm, but that's pretty much it. They sometimes have substructures like cilia and flagella for movement, but they're pretty simple organisms, and they're only single cellular. The more complicated eukaryotic cells, which we'll get into, evolved later, much later actually. We are considered eukaryotes. Even amoebas, which are single-celled eukaryotes, are eukaryotes, okay? They're more complicated than prokaryotes are. So, as I said, all cells have some features in common. So these are the things that prokaryotes have. They have a plasma or cell membrane. Those are synonyms of each other on the outside. It's semi-permeable. There's a cytoplasm within it, or cytosol you'll hear as well. Again, synonyms for all intents and purposes. You have DNA. DNA is the recipe book for making proteins, right? That's where your genes are. Prokaryotes have DNA, eukaryotes have DNA. And ribosomes. Now, prokaryotic ribosomes and eukaryotic ribosomes are slightly different. They're similar. They have the same basic job, but both prokaryotes and eukaryotes have ribosomes, okay? And that's kind of where the similarities in terms of structures end. So why are cells so small, right? Cells have to be really tiny. That's because tiny things are actually more efficient at diffusing materials across. And remember, diffusion is the movement of things from a high concentration to a low concentration across a membrane, whether they're using HELP, which is called facilitated diffusion, or they simply diffuse across from high to low across the membrane. Diffusion is the way that cells get their nutrients. This happens at a very regular rate, depending on the temperature of the external environment. Now, I'm going to show you in a second why this works this way, but we can actually quantify and determine which cells are more efficient by checking their surface area to volume ratios. So surface area to volume ratio is simply taking the surface area and dividing it by the volume. And you can see how the smaller cubes have a lower surface area to volume ratio here. I'm going to, if they have the same shape, so if we hold shape in constant, the smaller thing is always going to have a higher surface area to volume ratio. I want you to notice this is an improper calculation. That should say 1.2, not 12. So let's do this calculation. Surface area of a cube, right? That's the outside of it. It's the side, the area of one side, times 6, because there's 6 sides. We get 6. The volume of a cube is side cubed, which is 1 times 1 times 1, which is 1. The surface area to volume ratio there, 6 divided by 1, is 6. Let's take a larger cube, 5 by 5 by 5 cube. The total surface area is 25, 5 times 5 times 6, which is 150, divided by 5 cubed, which is 125. When you do that calculation, you get a 1.2 as the value, a lower surface area to volume ratio. So which of these two has a higher surface area to volume ratio? That's right, the smaller cube. If you take that 5 by 5 and split it up into tinier cubes, look, into all 1 by 1 by 1 cubes, what happens? We get a higher, or the same surface area to volume ratio. A higher one is the 1 by 1 by 1 cube. So it makes sense if you want to have something larger, it's more efficient if it is made up of a bunch of tiny things as opposed to one large thing, right? You don't see giant cells rolling down the street. So let's talk and look at a little bit about why that is. So if we look here, let's pretend that we have our two cells. We're going to have one giant cell here. I apologize for what this looks like because I'm drawing with my finger on a mouse pad, and a little bit smaller cell here. So let's say within, and I'm making this up, but it should hopefully get the point across. Within about 20 minutes, we allow for about this much diffusion to occur within this cell. Okay. And that means on all sides. I'm just drawing on 4 sides, but it would be, right, here's 5, all sides. But notice the core of the cell, core of the shape, has not received nutrients yet within 20 minutes. Same rate, so they're going into the same, that is demonstrated by the length of the line, same rate on this one. Notice that in 20 minutes, every part of the cell has received nutrients, right? There's no core at the middle that doesn't have them. So given the same shape, the smaller cell is more efficient. It's not that diffusion is occurring faster, it's just more efficient. So smaller objects are just going to be better at this in general, which is why cells have to be so small. Okay, hope that makes sense. Let's move to the next thing here. What the AP requires you to do then is actually calculate this stuff. Okay, so what we are going to do here, you're going to calculate the surface area to volume ratio of each object. Now in this case, they give you the surface area and volume of these shapes. They could make you calculate it also, the surface area and the volume, and determine the surface area to volume ratio. If you look on the AP reference table that you all get, you do have a part of it, pretty substantial part of it actually, that gives you all of the different surface area and volume calculations, or equations, excuse me, for different shaped objects. So you may actually have to calculate it. We will look at that in a second. I'm going to do a question that requires that as well. So let's calculate each of these. So I'm going to put up a picture here real quick. And that picture shows you how you would set each of these up. Okay? So in your space below, you can either pause it and copy down what I have, or you can try to do it on your own. And I want you to try to do the math here. Calculate the surface area to volume ratio for each of these shapes: the right circular cylinder, the triangular prism, the rectangular prism, and square-based pyramid. Okay? Go ahead, write them in your notes. All right, now what I'm putting up is the answers. So what you can see here is we have, for the right circular cylinder, you get 0.16. I just rounded to the nearest hundredth. I didn't tell you how to round, but let's say we do the nearest hundredth. Triangular prism is 0.25. The rectangular prism is actually the same as the right circular prism, which is 0.16, and then the square-based prism is 0.24. So my question is, which of these is the most efficient? The most efficient one is going to be the triangular prism, okay? So now we're going to actually do a question where I'm going to show you how you can use either the math to answer this, which will take a lot longer, or just your brain and what I told you about the rule. Smaller things, if the shapes are the same, are going to be more efficient at diffusion. So here's the question. A spherical bacterial cell has a radius of 3 micrometers, and the human egg cell has a radius of 100 micrometers. Which statement correctly indicates the cell that is more able to efficiently exchange materials with the external environment and provides a correct explanation? Remember, first thing you check yourself, the smaller thing is always going to be more efficient if the shape is the same, right? We're talking about a spherical cell and an egg cell. They're both spheres, basically. So the answer should be the bacterial cell, right? So it's not the egg, so we get rid of A and B. Now we're to C and D, so pick it. Because it has the smallest or the largest surface area to volume ratio. Remember, high surface area to volume ratios are more efficient. So the answer to this one is D, okay? But now I'm going to have you actually sit down and try to calculate the surface area to volume ratios for both cells. Now in order to do that, you're going to have to go back to this image right here and find the surface area and volume equations for a sphere, because that's what we're dealing with is a sphere, right? You're going to have to go and plug in. I'm going to put a snapshot of it once I'm done. So, if you don't understand how to do it, that's fine, but I want you to try to do it on your own first. Of both of them, you have to calculate the surface area of the 3 micrometer one and the 100 micrometer one. So you would plug in, right, 4 times pi times 3 squared, 4/3 times pi times 3 cubed, and then divide those two values, surface area divided by volume. Then you do the same thing for the 100, right? 4 times pi times 100 squared, 4/3 times pi times 100 cubed. You do that. So I'm going to give you a second to do that, okay? I'm going to pause. The video will pause. Okay, what you should have gotten, and I'm going to hold up my work, is for the surface area of the sphere, you should get 113.1, and for the volume of the sphere, you should also get 113.1, okay? Don't stress out that it's the same answer, it just happened to be that way in this case. When you do it with 100 though, the surface area is 125,663.71, and the volume is 4,188,790.2, I think 0, something like that, right? So now, which one's going to have the higher surface area to volume ratio? You can calculate it, but I can see which one it's going to be. The surface area, the 3 micrometer one is a 1-to-1 ratio. This has a much higher volume than surface area, so its surface area to volume ratio for the higher one is going to be lower, right? A smaller number. Therefore, again, we kind of show that the smaller one has a higher surface area to volume ratio than the larger cell does, okay? So cells need to be small. We're actually going to hit surface area to volume ratios a little bit more later as well. So as previously mentioned, there are 2 types of cells, main types. There's prokaryotic cells and eukaryotic cells. Prokaryotes are simple, they're bacteria, they are only single cellular, they are tiny, they're simple. Where prokaryotes can be single or multicellular organisms, and they're much more complicated. And we'll talk about why that is. And because of that, those cells can also be larger, and because they can be multicellular, you can get way more cells. You can get much larger organisms because of that. And here's showing you the difference between that prokaryote and that eukaryotic cell right there. Okay. And how much more complicated that prokaryotic cell is. And the reason it's that much more complicated, about 10 times larger too, has to do with what are called membrane-bound organelles. So it says here, prokaryotes are simple, no membrane-bound organelles or nucleus, simply circular DNA. That's important. The DNA is circular, meaning that instead of being a long single piece of DNA, it actually attaches on the other end. So it's a big circle. That's all, like a loop almost, that then is kind of jumbled up in the middle of it. it's hard to see here. But if you were to stretch this whole thing out, what you would end up getting is something that looks more like this. If we were to stretch all the DNA out, it would look like that, as opposed to a prokaryote or a eukaryote, which would have just one long piece that doesn't connect at the other ends. And if you're imagining like the like DNA, it would be much more smaller than this. But right here's that double helix structure that we kind of looked at when we went through macromolecules. It would look something like that. Whereas this is more linear, found in eukaryotes. Okay. Prokaryotes also have a cell wall on the outside, but that cell wall is made up of what's called peptidoglycan, which I'm going to get to in a little bit. When we talk about different types of organelles, even though it's not technically an organelle, the cell wall. So why are eukaryotes allowed to be larger and still exist? And actually, why are they more efficient? Well, it's because instead of having their cell membrane just do jobs, they have internal membranes that do jobs as well. Those internal membranes separate and sequester, right? That kind of means a similar thing. Sequester means to separate things away from each other. Different chemical reactions. So you're allowed to do more like specialization within a eukaryote. That makes them more efficient, and that makes them more, because of that efficiency, they're able to be larger and more complicated. That's a question that AP loves. What is the benefit of organelles? Increases the surface area internally, separates reactions, makes them more efficient because of that. So again, back to that image I showed you before, you can see the membrane-bound organelles within this and how much more complicated it is on the inside. It allows for more surface area to volume ratio, right? More surface area here around with these structures here and internally. So what are other things that cells do to allow them to have increase the surface area while keeping a larger volume? So like, for example, you have neurons in your body. They're your nerve cells. The longest neurons in your body are over 3 feet long. One cell, 3 feet long. But why is it allowed to exist then? Because it's so thin. It's so incredibly thin. You can see this axon right here is really thin. That's the thing that's 3 feet long. And because it's thin, it still has a really high surface area to volume ratio. Some cells have folds and microvilli along their membranes. Like here, like these are digestive system cells, like small cells found in your small intestine. They have these projections. It increases their ability to absorb nutrients and materials into the cell there. And again, that's a structure-function relationship, because what is your digestive tract's job? It's to absorb nutrients, right? All of these structures increase efficiency. So here's looking at a eukaryote. Now we're going to focus on those, much more complicated, and we're going to look at some of those substructures within it, right? Those membrane-bound organelles. So let's see what we got here. There's 2 types of eukaryotic cells that we focus on, though there's actually more than that. But the 2 that they tend to focus on are the animal and the plant cells. And you may remember this back in the day when you're even in middle school or freshman year, you learned about what are the organelles that are different between plants and animal cells, right? So a big difference is plant cells, similar to actual prokaryotes, they have a cell wall. That cell wall is made of different material. We'll get into that. That plant cell has a large central vacuole for storing water, which is very important in plants. And probably the most distinguishing characteristic is the presence of chloroplasts, which make plants plants, right? Chloroplasts are organelles that allow for photosynthesis to occur, which is our main, thing we think of biologically when we think of what plants do. They don't have true lysosomes or centrioles. Again, don't worry about the centrioles that much. They're involved in mitosis for organizing, pulling apart the chromosomes during mitosis. But plant cells don't have those structures, but they do have a cell wall and chloroplasts. Animal cells have no cell wall. They never do. They have a lot of tinier food vacuoles within them, no large central water vacuole. They do not have chloroplasts because they're heterotrophic. They have to go and get their food from outside of it. They do have centrioles, which are the parts that organize the fibers for pulling out chromosomes apart. and they have lysosomes for digesting the food that they consume. That's the job of lysosomes. So that's kind of the difference between plant cells and animal cells. There are also fungi, right? That's another type of eukaryotic cell, and they're weird. They're like an animal-plant cell hybrid almost. They have cell walls. They're made up of a different material, again, than both bacteria and plant cell walls are made out of, but they are heterotrophic because they have to absorb the nutrients from the environment. They don't do photosynthesis. So those are the 2/3, if you count fungi, types of eukaryotic cells. Let's talk about those substructures now. So there's two different types of substructures that we look at: the non-membrane bound structures that both prokaryotes and eukaryotes may have, and then the membrane bound or true organelles. So let's start with the non-membrane bound structures first. The first is the ribosome, and ribosomes are vital. They are the protein factories. They're like little chefs that read mRNA, which is the copy of the gene, and assemble the structures, the primary structure of the protein. So their job is to take mRNA, read it, and put together in order the amino acids that make up the primary structure of the protein. And remember, once that primary structure of the protein exists, the protein will start to fold due to R group interactions and form that primary secondary, which is alpha helices and beta sheets, tertiary, and sometimes quaternary structure. Right? So ribosomes are protein factories. They are chefs. They build proteins. Sometimes they are free, meaning they are found free floating in the cytoplasm, and other times they are bound to another organelle, which we're going to get to in a second. Remember, eukaryotes and prokaryotes have ribosomes, but the prokaryotic ribosomes are a little bit smaller than the eukaryotic ones. The next thing is the cytoskeleton, which you maybe have heard of the cytoplasm, but the cytoskeleton includes all of the fibers and networks that are within the cell that give it its structure. It allows it to kind of move if it's a motile or mobile one. Sorry, this didn't work. I'm going to insert the video in a little bit. It also acts as a place for the walking, which sounds crazy. There's these special proteins called dynein and kinesin molecules that grab packages, vesicles within the cell and move them throughout the cell. And it looks like they're walking, but they're simply just proteins changing conformational shape as ATP is being hydrolyzed and used. Okay, it's showing you that in this video right here. Here's kind of showing you that again. You can see that dynein and kinesin molecule walking along one of the cytoskeleton fibers. There's different types of fibers within the cytoskeleton. There's thin filaments, thick filaments, there's intermediate filaments, there are microtubules. There's all different types of things, but all of them, their job is to provide structure for the cell, help the cell move, and also allow and act as the roads in which things can move throughout the cell. Okay, so that's the point of the cytoskeleton. And a branch of the cytoskeleton, which is involved in movement, is the cilia and flagella. Cilia are a bunch of small little hairs on the outside of the cell, and then a flagella would be one or two large projections. They all move, and their job is to kind of move the cell around. In eukaryotes, we also have some adaptations, like the famous eukaryotic flagella is found on sperm cells, but we have cilia like in our trachea. Even though it doesn't allow the cells to move around, it moves stuff on the surface of the cells like mucus, so it allows us to function. Okay, this should say, this kind of got cut off here. They're locomotor appendages in some types of cells. Locomotor means movement. Not every single cell has these, but it's a commonly seen non-organelle structure that is made up of microtubules, which are part of the cytoskeleton. And then the last one's going to be the cell wall. So cell walls, again, not found in animal cells, only in plants, fungi, and then in prokaryotes, but different types. The cell wall is an extracellular structure. It's on the outside of the membrane. You can see the membrane at the bottom with that lipid bilayer with the phospholipids, the hydrophobic tails, hydrophilic heads. Plant cell walls are made of cellulose versus bacterial cell walls, which, as I'll talk about here, are made of peptidoglycan, slightly different, but they still have the same job, which is it provides this rigid structure on the outside of the cell, It does allow for certain things in and out, like if something's really big, it can't get across that. But really its point is to provide structure, like an external type of skeleton for those cells. Bacteria have them as well. As I said, they're made of peptidoglycan, and we actually classify bacteria based on the type of cell walls that they have. There's gram-positive, gram-negative. You don't need to know that for the AP exam. That's more of a college level thing. But they do have different types of cell walls. All right, so now let's talk about membrane-bound organelles, the ones that allow for these eukaryotic cells to be much more complicated than their prokaryotic counterparts. The first one, which we all know, is the nucleus. That's the genetic library. So this stores the DNA within it. It has, because the DNA and the regulation of DNA, which genes are turned on and off, happens within this structure. And the proteins are the things that do everything, right? And DNA codes for those proteins. We consider this the control center of the cell. It basically dictates within it what proteins are made and which ones are not, okay? So what happens is that DNA gets copied and turned into mRNA, and that mRNA then, sometimes we'll talk about this, but it gets processed, and then it leaves the nucleus and it's read by ribosomes, where the ribosomes, which are outside of the nucleus, eventually assemble the primary structure of the protein. So the nucleus, genetic library of the cell. The next one is the endoplasmic reticulum. We consider this like the biosynthetic factory, and there's two types of it that are continuous with each other. There's the rough ER and the smooth ER. The rough ER is called rough because it looks like it has a rough texture to it due to the ribosomes that are attached. These are the ribosomes that are making proteins that their job is to get shipped to other places within the cell or out of the cell, okay? If it's a free ribosome, which is floating in the cytoplasm, those ribosomes' jobs are to make proteins that are destined for the cytoplasm. But these are destined for different organelles and for the outside of the cell or the cell membrane, okay? So that's the purpose of the rough ER. The smooth ER does not have ribosomes, that's why it has a smooth appearance, and its job is to mostly, for all intents and purposes, do really 2 to 3 main things. One, it detoxifies poisons within the cell. Its job is to take out bad things within the cell and detoxify. Our liver cells, for example, because they're a detoxifying organ in our body, have a lot of smooth ER within them. And if people are like drink alcohol a lot or take other types of things that need to be detoxified, their networks of smooth ER are more and more dense. It also helps to synthesize lipids for the membrane, so it helps to send in cholesterol, but like the membrane, it sends lipids to the membrane, enhances the size of the membrane. The rough ER can kind of do that a little bit too, and it sends vesicles to it, which we'll get into in a second. And it also can store calcium within it as well, which can be released when needed in all cells, but it's very commonly seen as an important physiological mechanism in muscle cells to allow muscle cells to contract. You don't have to know that for the AP Bio exam, though. I'm just giving you information. The next thing is the Golgi apparatus. This is the shipping and receiving center. So what is the relationship between the Golgi apparatus or the Golgi body and the rough ER? So what happens is the rough ER will receive proteins that are meant for shipping out initially. So they receive the first signal to say okay, we need to order these proteins. And their job is to get them, let's say, to the membrane, okay? What happens is it takes the proteins and makes them, and then it ships them off in a vesicle to the Golgi. The Golgi's job is then to reorganize it and put everything together that needs to go to the same place in one single vesicle package, and then that package can be shipped out to its proper location. The analogy that I like to use for this, and you're going to have to write this in your notes, so pay attention. You may have to rewind it if you don't get it the first time, is like an Amazon warehouse or an Amazon ordering center, and then UPS. Now, Amazon now has its own shipping company, but let's pretend they didn't, like back in the day. And sometimes they still use UPS to ship their stuff. So Amazon receives the order, and then someone or machines or whatever goes and grabs all of the things that you ordered and puts them together, right, for your order. That then goes to UPS, the Postal Service. The Postal Service will then take everything that needs to go to the same place, like in your neighborhood, and put it on one truck and then ship it to that location, right, to your house, to that neighborhood. That is the analogy. So the rough ER is like that Amazon warehouse The ribosomes make the proteins, put it in there. Small packages go to the Golgi. The Golgi then organizes all those small packages that need to go to the right place and puts them in their proper location. So hopefully you never forget that. This is actually showing you that whole process. They're showing you the vesicles here, which are these small little packages, right? 2 different types of vesicles. Here's the rough ER. So the mRNA leaves the nucleus, goes to the ribosome then reads it, spits the protein into the rough ER. The rough ER packages up some proteins, puts them together, then that gets shipped via vesicle to the Golgi. The Golgi decides everything that needs to go together in one place, it then ships it out, and then it ends up sending that vesicle to the membrane or wherever it needs to go, okay? Remember, free ribosomes would be like little ones sitting in the cytoplasm. Those are for proteins that are destined to just stay in the cytoplasm. And as we go through the course, we'll talk about different examples of that. Lysosomes are important for carrying out intracellular digestion after phagocytosis, which means phagocytosis is cellular eating. The cell will engulf something like a food item, and then it needs to be broken down to its component macromolecules. So remember, lysis means breakdown. So lysosome is a breakdown organelle. You see a lot of these in white blood cells because those are engulfing pathogens all the time. But that's the main job of lysosomes. But another thing that they do is that if there's like organelles that are not functioning or that are getting too old, the lysosome will fuse with it and break down, right, those organelles so that we can reuse those building blocks to maybe make other organelles or other proteins. And lastly, its job is to do apoptosis. Apoptosis is also known as programmed cell death. So when a cell has reached the end of its life or something has gone haywire within the cell, there's a process called apoptosis that gets triggered. And if this process gets messed up, you can have cancer and things like that, which we'll talk about later in the year when we get into that. But the lysosome will literally release its digestive enzymes into the environment, into the cell. And it will cause the cell to form these things called blebs, which are these like impinchings of the cell. And that means the cell is inevitably going to die or that it's been destroyed. So that's an important mechanism for getting rid of cells that have gone haywire, either because viruses have infected them or they've become cancerous and are growing out of control. So apoptosis, autophagy, and digesting are all roles of the lysosome. Remember hydrolysis, lysosome, breaks down. Peroxisomes are complicated. They don't ask about them that much, but I like to talk about them because you may learn about them if you do major in biology in college, or even if you don't and you take another biology class. Peroxisomes are organelles that both manufacture hydrogen peroxide and also break down hydrogen peroxide. Hydrogen peroxide is a really, really nasty molecule, but it also can be used for good. It is a very strong oxidizer. It rips electrons off of things. So actually, it synthesizes it synthesizes peroxide so that it can help to oxidize fatty acids. It breaks down fats. That's a good thing. But also, because that peroxide needs to be gotten rid of because it starts to actually oxidize the cell itself, which is a problem, the peroxisome also needs to produce an enzyme called catalase, which we're actually going to do a lab on, which its job is to break hydrogen peroxide down into both water and oxygen gas, which are safer, obviously, molecules than the hydrogen peroxide is. So that's the job of peroxisomes. They help to detoxify poisons by oxidizing them and also help to digest fats by oxidizing them, but that peroxide needs to be dealt with, which is why they have an ability to break the peroxide down as well. Mitochondria, the powerhouse of the cell, right? Mitochondria are found in all eukaryotic cells, both plant and animal, right? A lot of people think, there's a misconception that mitochondria are not found in plants. Like mitochondria in animals and chloroplasts are found in plants. No, mitochondria are found in both. Because if you're a plant, the mitochondria, the job of photosynthesis is to make sugar. The job of the chloroplast, in plants, the job of the chloroplast is to make sugar through photosynthesis. That sugar then needs to be broken down through cellular respiration. That's what happens in the mitochondria. So mitochondria, as I said, are found in all eukaryotic cells, and they are where cellular respiration occurs, and the job of that is to make ATP using oxygen gas. Interestingly enough, the mitochondria, and we'll talk about why this is later in the year, it has two membranes. It has an outer membrane and then an inner membrane that is highly folded. Now I want you to think about what the job of the mitochondria is. I just said it. And why would the folds help this process? The folds increase the internal surface area to volume ratio, right? And if the job of the mitochondria is to make ATP, that means that the chemical reactions, which we're going to get into later in the year, the chemical reactions that do that process are going to become more efficient. You can do more of them because there's more surface area to do those chemical reactions, okay? So it's an example of a surface area to volume ratio situation, okay? And lastly, there's chloroplasts. Chloroplasts are the light-capturing organelles. It's a specialized member of the plastid family. There are a bunch of different types of plastids, the chloroplast being the most famous one. Its job is to do photosynthesis. It takes light, it makes glucose using raw materials, CO2 and water from the environment. And chloroplasts are found in leaves and other organs within the cell that are involved in doing photosynthesis. All right. That is the entire part. Those are all the different ones. So let's look at these questions. Which cell would you most likely expect to find lysosomes in? Let's look at this and pause it. Try to answer it. The answer is C, macrophage that engulfs bacteria because it has to digest that bacteria. Which cell would you expect to find lots of smooth ER? The answer to this one would be ovarian cells that produce estrogen. That's a steroid hormone. That's a lipid, right? Which would you find lots of bound ribosomes found on the rough ER? The answer to this would be B, the pancreatic cell that manufactures digestive enzymes that need to be shipped out. How about where would you expect to find the most mitochondria? The answer to this is A, the muscle cell with the thigh muscle cell of a long distance runner because they need lots and lots of energy. Okay? Cells of the pancreas will incorporate radioactively labeled amino acids into proteins. This tagging of newly synthesized proteins enabled the researcher to track the location of these proteins in a cell. In this case, we are tracking an enzyme that is eventually secreted. Which of the following is the most likely pathway of the movement? What is the order? Remember, it goes ER to Golgi to vesicles that fuse with the plasma membrane. All right, that is the lecture on organelles and cells. If you have any questions, come to class, ask me.